over the years.
Well, good morning, everyone. Thank you for coming to I believe it's the third of the lecture series, Marine Science in the Morning. So we do this every 2 weeks. This will happen in another 2 weeks. So I hope you can join us for the next lecture. And we're proud to have our best aquarium volunteer and professor emeritus from FAU Harbor Branch Oceanographic Institute. And we're excited to hear him give this talk today on the Oculina Reef. Thank you, John.
Thank you, Lisa. If you've never been here before, right in this hallway over here is an exhibit about the Oculina coral reefs. And out of that 40 years, 49 years that I worked at Harbor Branch as a marine biologist, a good part of my research was on the Oculina. I published some 30 papers, scientific papers, describing, mapping the reefs, describing the geology, the biodiversity, the fish, the impacts from fisheries, and conservation efforts.
There you go.
So offshore, off the east coast of Florida, we have these rocky ledges called hard bottom communities. And one of the tanks out here is hard bottom community. This is looking at North Beach, North Hutchinson Island. Up the far end is Port Pierce Inlet. And this dark area are hard bottom rocky ledges. So we have these rocky ledges that provide habitat, but these are not coral reefs. You don't get coral reefs until you get down to West Palm Beach and to the south. The water is too cool up here for a typical coral reef. So these near shore hard bottom habitats, the first reef occurs between about 10 feet and 30 feet, goes out about a mile. Then you go further offshore, you get another reef system at 30 feet, and then another one at 60 feet and 90 feet. And these are covered and provide habitat for all kinds of sponges, gorgonians, invertebrates, lobsters, fish, soft corals. This is right off here, right off Port Pierce. But there is some coral that grows there. It's not a coral reef. These are individual coral heads. And this is the Oculina vericosa. In the tank down the hallway, you can see what this coral looks like. And in the shallow water, it is brown, forms little heads about 1 to 2 foot in diameter. And unfortunately, they get hung up with fishing gear and nets, people throwing cast nets. So this is the coral called Oculina vericosa. In shallow water, it is brown just like any shallow water coral. Shallow water corals have a symbiotic relationship with the animal and plant and algae that lives in the tissue, as well as the microbiome. Just like we have a microbiome in our gut, they have a microbiome surrounding the animal. This is a close-up of the coral. These little circles with tentacles are the animal portion of the coral. So that animal portion makes CO2 and excretes waste. And those little tiny dots are the algae called zooxanthellae that live in the tissue of the coral. So the algae produces oxygen and food that the animal portion eats. So the symbiotic relationship that all shallow water corals have is what keeps the corals living. So unfortunately, in the last few years, as temperatures rise, global warming, we've had some huge records of temperatures in the Florida Keys and throughout all the reefs. Some of the temperatures in the Florida Keys have gone up to over 100 degrees. So when these shallow water corals are stressed, they expel the zooxanthellae. We're not really sure why or how, but these zooxanthellae are single-celled algae about the size of red blood cells. Red blood cells, very, very tiny. They expel them, and if you've heard of coral bleaching, the coral becomes white. There's no algae in it. Now, this algae floats around in the water. If the temperature drops down soon enough, the coral can reabsorb the zooxanthellae. If the temperature doesn't drop, in a few weeks, the coral will die. We've seen massive die-offs for the Keys and the Caribbean in the Great Barrier Reef due to global warming and coral bleaching. Now, for some reason, we really don't understand why, Oculina coral are one of the few species that can live with or without zooxanthellae. Now, there's other deep water corals that only live in deep water. These are called cold water corals that can grow down to 2,000 feet. But they are also white, but they cannot live in shallow water. So the Oculina is about the only species that can live both shallow water with zooxanthellae in deep water. It may be the savior of our reefs in the future. This is a close-up of the deep water coral. In deep water, they get very spindly. They're very fragile. If that was growing in shallow water, it would break up in the wave surge. These can grow up to be 5 feet tall. That coral there from top to bottom is about 10 feet long, probably 6 feet tall. And these form mounds that the coral grows on. So right at the shelf edge break, in about 300 feet of water, you find these mounds of coral, and the corals living on the sides and the top. This is a fathometer profile. This is what the depth looks like from 27 to 32 degrees. This is Port Pierce. You go offshore with your fathometer, your depth finder, right at that shelf break, where it drops up steeply, you have these 100-foot tall coral mountains. It goes all the way up to Cape Canaveral, and now they're trying to go up to Daytona Beach. The Oculina variegosa is also called Oculina. Ocu for ocular, eye, and ina for little eyes. So that picture I showed you before, they look like little eyes, Oculina. So they grow throughout the Caribbean, but only here, off Port Pierce of the Daytona, do they form these deep water mounds and coral reefs. Nowhere else on Earth. Nowhere else. So our first studies where we discovered these reefs were in the mid-1970s. We were doing a survey with the Johnson Sea Lake submersible, just doing general surveys of the continental shelf, seeing what was out there. We knew very little. And so we did dives from 100 feet deep out to 1,000 feet deep. Each little bar is an individual sub-dive. So that took about 5 years of us going back and forth, documenting the shelf. And what we discovered is Oculina mounds occurred in that the shaded zone. That's from Fort Pierce up to Cape Canaveral. That's how much we knew at that time. And later on, we found out that they were fishing, new types of fisheries were coming down, dragging trawl nets through the reefs. And so I petitioned the South Atlantic Fishery Management Council to make it a marine protected area banned bottom trawl. We had 3 ships at that time. This is a 200-foot ship, the Edwin Lake, and we also had the Stuart Johnson. And they carried the submersible on the back of this huge A-frame. We drop the submersible in the water, release it, and then the sub could swim freely. Sub carries 4 people. The front is a spear, an acrylic spear 6 feet in diameter, 5 inches thick, and it had a crush depth where if it went too deep, it could go to 9,000 feet before it would crush. But we only went 3,000 feet, gave us a 3-to-1 safety ratio, and like the Triton sub that didn't know what they were doing. This ain't front of the sub. We had all things here. We had cameras, video camera, still camera. There's this manipulator arm. It had a clamshell grab for picking up sediment and soft animals. Fingers to pick up rocks. Suction hose. Right here, they could suck up samples, and they'd go in a Lazy Susan and the buckets that were located here. All of this was built at Harbor Ranch. They built the submersible, developed the submersible by Edwin Lane, who was the inventor, co-founder of Harbor Ranch. We also mapped the bottom. This is an AUV, an automated underwater vehicle. They could map the bottom with 3D imagery showing a topographic map at the bottom. It's not a picture, but it shows a 3D image called multi-beam sonar. So you get kind of like a topographic map of the mountain. This is a multi-beam sonar, and all these little brown bumps are the individual coral reefs, 60 to 100 feet tall. The biodiversity of the reefs are incredible. I spent many years studying this. 70 species of fish, mollusks, crustaceans, echinoderms, polychaetes. All these animals live in that bushy coral and provide food for the bigger. 70 species of group, practical more than most of the work on the fish. Gag grouper, scamp grouper, snowies, Goliath grouper, snappers, migrating fish like amberjack, mackerel, wahoo, tuna, little tunny, ocean sunfish, manta rays, sharks, and all the little reef fish that make food for the bigger fish. So this is like the base of the reef. We're out here with the sub looking toward the reef. And these are all different types of grouper. We've got scamp grouper, gag grouper, snowy grouper. What's incredible that this exhibit out here shows that the gag and scamp grouper go to the deep reefs when they're sexually mature. And they're 15 to 20 years old before they're sexually mature. They go out here and spawn. Female swims up, releases her eggs. Males swim through the eggs, releasing their sperm, making little fish, fertilizing them. These larvae, small fish, what we call stream. Many of them come inshore and come into the inlets and end up in the Indian or the lagoon and spend their lives in the grass beds and mangroves in the lagoon. They're pretty incredible. These reefs that are 30, 40, 50 miles offshore, the grouper that live out there rely on the grass beds in the lagoon. When these grass beds die, we're affecting the fish and fisheries offshore. Yeah, these guys are like 3 foot long. Speckled pine, 3 foot amberjack, huge schools of amberjack. I'd be diving down there and be 100 amberjack just circling, going round and around in the Warsaw machine. That was about a 500-pound Warsaw group. He came down to visit me a number of times when I was diving on the reef over a couple of years. And one time we were in the sub and he started swimming away from the reef and we decided to follow him. So we followed him away from the reef. It's a flat sand bottom. We wanted to see where he was going. We went about 2 hours before the Pierce up to Vero and came up to a new reef that we didn't know about. He knew where that reef was and he would go in there and he showed us where it was. This is a tiger shark. He visited me during a lockout dive one time. This is about an 8-foot mola, ocean sunfish. He had a little scar on his forehead, and so I knew it was him. He was out there 3 to 4 years. He would come down to the table where I was doing my research. One of my first studies, we knew nothing about deep water coral reefs, the ecology, biodiversity. One of my first studies was to collect samples with this fine mesh bag, about 1 millimeter mesh, put it over a coral head about the size of a soccer ball, and collected everything on and in the coral to see what lived there, what was the food web, what was the biodiversity. And a coral head that size, I counted 2,000 animals, crabs, worms, little fish. It's incredible biodiversity. Some of these animals are in the exhibits out here. Not these animals, but brittle stars, giant red brittle star that's in the coral tank out front. Pencil urchins, giant red starfish. We had 230 species of mollusks. That's crab, snails, bivalves, and even cephalopods, squid, which is a type of mollusk. Most of these are pretty small. They're living on in the branches, little habitats for them, scallops, bubble shells, top shell. Oh yeah, it's a very beautiful shell. Orange shell, and it kind of wraps itself around the coral and actually holds the coral together, helps support the coral. This is a squid. We did a dive. This is from a lockout dive I did, 70 meters, 1977, and the whole bottom was covered with these dead squid. Now, squid after they mate die, and so apparently it was a big spawning event. The whole bottom was covered with squid. I came back the next day and they were all gone. They were all gobbled up. 50 species of decapods, crabs and shrimp. Galloped crabs, hermit crabs, porcelainic crabs. Be a shrimp, little tiny slipper lobster about this big. So we studied the physical environment out there and found that temperature was quite variable. When you had upwelling, cold water coming out of the Straits of Florida would come up on the shelf, and the temperatures could drop down into 40 degrees. And other times when the Gulf Stream kind of going over the top, it would drop down. Gulf Stream was raised the temperatures up to the 70s. There's no shallow water coral that survived that. It couldn't survive 40 degrees. Light transmission of only 0.3%. It was not black, it was twilight. After sunset, everything's kind of bluish. We had an incredible team of engineers. John Holt here essentially developed and engineered and built these submersibles, the Johnson Sealing submersible. This is a time-lapse camera we put out on the reef. This is pretty cool. So we had time and took a picture every minute over 24 hours. Set it up in front of a coral head And a series of photos during the day, see all these fish swimming around. As soon as it became dark, the fish disappeared, and all these little animals crawled out on the tips of the branches, little shrimps and crabs and mollusks out there getting food. And the first of the sunlight, they zipped back into the coral and the fish started swimming around. The temperature, current meters. This is pretty neat. So this 80 meters, 300-foot reef. This is the temperature. So down here for each month, you're down in the 40s, 45 degrees. You get up to 70s during the warmer periods. But what's interesting, the coolest time is like July and August on the deep reef. And what happens in the summertime, you get the southeast wind blows on the shelf. And due to the Coriolis effect, the southeast wind is actually pushing the water off the shelf. It's 90 degrees to the angle of the wind. It's pushing the water off. So you're looking in the Gulf Stream, actually the water's lowering on the Florida side and higher on the Bahamas side. And so what happens, it kind of pushes down and forces the bottom water up on the shelf, so you get this cold water upwelling in the summertime here. This is how we did lockout dives. The back of the sub was a pressure chamber, like a recompression chamber, and 2 people in the back. They put on full face masks. You're trying to suit up in 3/4 inch wetsuits and stuff in a little chamber. And then we had about a 75-foot hose that provided the gas mixture to breathe. So 300 feet, you can't breathe there. You pass out. So we had to have a special mixture of 10% oxygen and 90% helium. And this is a cross section. The front of the sub, where the pilot and observer are. The back of the sub, the front part stays at 1 atmosphere. You just go in there in a shirt sleeve, and they pump in oxygen and scrub CO2. The back is the dive chamber. So we would pressurize the dive chamber, open up the hatch, and crawl out the bottom and dive and swim around the ship. After the dive, we would close the hatch, the sub would come up on the ship. And then at that time, we had a larger chamber at below deck in the ship. So the sub would come up. We're still pressurized in the back. We would make the sub to this chamber. I could go down here, and there was bigger, so there were bunk beds, there was heating, we could have food, we could go to the bathroom. And for a 30-minute bottom time, I had to decompress 5 hours. We were coming up really, really, really slow. Came up too fast, you had decompression sickness, bubbles, the nitrogen in your blood would bubble out. My other experiment was the growth rate of the coral. Nobody knew how fast this coral grew. So I put coral heads on a table 300 feet deep. And this coral I stained with a special dye of native paint. And the white part's the growth over a year. That was 6 centimeters. That coral only grew 1.5 centimeters a year. To grow a coral head that's the size of an SUV, we're talking centuries. So what are the main impacts now? The primary impact is from bottom fishing, trawling, or rock shrimp. Very small fishery has done an incredible amount of damage to the reefs. Other damage can be from long lines, fishing weights hitting the coral. It's very fragile. Fish tracks and anchoring, it primarily is trawling. Found nets wrapped around the reefs, trawl nets. It's only a handful of fishers that fish for rock shrimp that are out of Cape Canaveral. One fishery, one restaurant up there. That coral there is probably about 10 feet from the top to the bottom of the photo. You're looking down on it. It's probably about this tall. That's the same area. That picture before I took in 1978. This is the same site in 2001. After trawling, right off Cape Canaveral, 300 feet deep. That's the exact same pinnacle. There was only one mound out there where I took that first picture. I couldn't mistake it for anywhere else. There's one mound. This is the same spot. What it looked like before, 1970s, 2001. What happens, these are transects that show the loss of coral between the 1970s and 2000, when they finally closed off the fishing, closed off the area of fishing. But this is Cape Canaveral, Cocoa Beach, and Valley. Is how much coral was lost in that time. This is what happens when you lose coral, and you just have rubble. And in intact reef, you have all these gag and scamp grouper. On an unconsolidated reef or rubble, dead reef, nothing lives on it. So we did some restoration, putting out these reef balls. An exhibit out here shows some miniature ones. These are about 3 foot tall concrete structures, provide habitat for the coral to settle and grow on, and immediately fish started using it. So it's viable, but it's very expensive and not easy to put out. So what protection are we doing now? What have we done for protecting these reefs? 1980, I nominated them to be a marine protected area. 84, they finally designated the Oculina HAPC, which means Habitat Area of Particular Concern, which is the type of marine protected area, which bans destructive fishing here. 2000, finally got the council, the Fishery Management Council, to expand it up to Cape Canaveral. It made it 300 square miles. Then later on, I discovered the reefs actually went all the way to Daytona. In 2016, it was expanded to Daytona. So here we have the original HAPC from Fort Pierce, that little box of Sebastian, 1984. In 2000, it got expanded up to Cape Canaveral. And then recently, in 2011, I discovered the reefs actually went from Cape Canaveral all the way up here to Daytona. And that red zone, the red box, are where the Oculina reefs are up there. But this it shows those reefs where this little green area is where we mapped it with multi-beam sonar. So the map on the right shows the typical bathymetric contour map that we use to find the reefs. Each of those circles, it represents a coral mound, 20, 30 meter tall mound. So we figured that 1, 2, 3, 4, 5, about a dozen mounds in that box. But when we mapped it with multi-beam, we actually realized there was like 100 individual mounds that they just kind of show together in the style map. So I proposed that to be protected. The red line through the eastern line of that area to be protected out to 100 meters. So that would give a good buffer. If you were trawling east of that, you wouldn't accidentally hit the reefs. It would provide a buffer between poachers, and also the Coast Guard said it was a good idea to put the line, well east of the reefs, so they could, have surveillance and enforcement. Well, the fishermen, the rock shrimp fishermen last year that had submitted an amendment to allow them to fish within that outer zone of the northern area. They put an amendment to allow shrimp fisheries inside of this zone that would impact the coral there. Which seems rather odd since the purpose of the MPA originally was to protect the coral from the fishing, and now the fishers are saying that's their historical fishing grounds, and they have a right to drag in there. Their line go right over the edge of these reefs, causing sedimentation, land on the coral, will kill the coral, kill the planular larvae. It also is known to cause coral disease, stress from long-term sedimentation. So the yellow is actually accepted now. So the yellow and the red is the new Oculina HEPC. Those are the restrictions. People are allowed to surface fish troll out there, but not bottom fish, not use long lines or anything. So again, the recent events, the council extended the HAPC in 2016. The rock sugars in 2024 proposed Amendment 10 to the Fishery Management Act to allow the shrimp fishery to have access to the HAPC, which would set a very bad precedent if they get this. Public outcry stopped the amendment last year, not permanent though. You're looking at it again. 40,000 people wrote in about this. But unfortunately, the shrimpers are on the South Atlantic Fishery Management Council. They control the council, and so they're pushing for a new amendment to allow them to fish within the day. So what can you do? Is contacting people that no, we don't want to. So thank you very much. Questions, we're going to show it. A movie or a documentary, a short documentary. I had a summer intern a number of years ago. And I told her, I said, I have all these historical photos and videos. I want you to put together a short documentary telling about the reefs, about their discovery, conservation efforts. And so she and Brian Cousin, our videographer at Harbor Branch, and I put together this little video clip. Do I have to press something?
Thanks, Helen.
Nice. Do I do something? I see that. Thanks. Your volume is somewhere.
It's working.
Parcels and remotely operated vehicle in this cord. Scientists from the Harbor Branch Oceanographic Institution discovered the deepwater Oculina reefs in 1975. These eerie Deepwater Oculina reefs are similar to their shallow water cousins, with one distinction. The deepwater Oculina corals lack zooxanthellae, microscopic single-celled plants that live inside of shallow water coral, converting sunlight into food, a biological process known as photosynthesis. Deep Oculina coral capture plankton and nutrients from the rapidly flowing Gulf Stream. The Oculina reefs are located 200 to 300 feet below the surface, anywhere from 60 to 50 miles offshore. When the Harbor Branch submersibles first dived in the reefs in the mid-1970s, scientists aboard witnessed an unexpected diversity of life located on and around the Oculina reefs. Special form of diving, when it's log-out diving, when a diver actually swims out of the JSL submersible, allowed scientists like John Reed to study the Oculina reefs firsthand.
Back in the 1970s and early 80s, we used lockout diving, where we're actually tethered to the sub and breathe a spatial gas mixture of helium and oxygen. We used this to dive down to 300 feet deep. This allowed me to do most of my experiments that I couldn't have done
What's happening to the volume. And this is where they're recovering the sub. There again, this is one area that's been crushed to rubble. The one thing we are seeing, there's coral growing back. The little patches of coral. The last 10, 20 years, I'm starting to see corals this size starting to come back. If you're going to keep rolling out.
Bycatch of the trawling is just incredible.
Yeah.
It's like for every 1 pound of shrimp that they catch, between 3 and 5 pounds of other marine species are also captured for 1 pound of shrimp.
Now, trawling, shrimp trawling is fine in places like in the Gulf of Mexico where we have flat sandy bottom. You get your red, your pink shrimp, your white shrimp. That's fine, but not on live bottom habitat, and that's a problem worldwide.
No, I think it's around on the Lucies.
We just had another place and put it back where it was.
Now, if the trawlers harm the reef, doesn't it also in the long run kill their chances of getting the rock shrimps?
Well, the rock shrimp like rubble.
Ah.
That hurt the rock shrimp, but it's going to kill any other fishery. Let's put it back up where we were. Which is a snot. That's right. There's about 3/4 there. That's, yeah, through birth. Greg Gilmore did a lot of work on the stamping day through further breeding and population structures. The amberjack just kind of follow the shoreline, the coastline, and we just go from reef to reef. It's like hundreds of them come down, feed on the reefs, and then just move on. Amphibians, the whole bottom would be covered with those amphibians, small pink fish. And that is the primary food source for the larger grouper. Rachel fish. There's the Link Foundation, at Link, the primary founder of Harbor Branch Oceanographic Institute, along with Seward Johnson in the early 70s. That's the fish I'll look up to the other reef there.
How big is that fish?
I'm sorry about the audio, but there you